This portion of the chapter will cover some other ways to make alkyl halides, and this time we're going to spend most of our time talking about how to make alkyl halides from alkenes without actually destroying the alkene. In other words, we're going to create a functional group. Taking advantage of the presence of the alkene, we're going to create a second functional group in the molecule. That is, the halogen will be introduced and the alkene will still survive. The first thing we need to know is what we mean when we talk about the allylic position, because this is the position where we're going to be able to introduce the halogen. And if you look at these molecules, they're numbered, and what we mean when we say allylic is one carbon away from a carbon-carbon double bond. So the way this is numbered, the carbon double bond is between 4 and 5. So carbons number 6 and 10 and 3 and 7 are all one carbon away from the double bond. So all of those would be allylic positions. Now, about this particular molecule, you see there's symmetry in here, and so I can't really tell the difference between 6, 10, 3, or 7. They're all identical in that sense of being allylic, and because of symmetry, all identical, which is not true of this molecule. Alright, so here the double bond is between 5 and 6, the way this is numbered, and 4 is allylic, 8 is allylic, and 1 is allylic, and this has, this, by the way, these numbers have nothing to do with numbering of the chain. I just let the software number them. 1 doesn't look at all like number 4, even though they're both secondary. This one is next to a methyl, and this one's next to this other branchy stuff. And of course, number 8 is primary. So those are 3 uniquely different allylic positions, even though they're all allylic. In this molecule, we have some symmetry. 1 and 4 are both allylic positions, and I can't really tell the difference between those. At least in principle, I can't. And number 7 is also allylic, and it's different from 1 and 4. Now, technically, I kind of hedged a little bit, I'll admit it. If you really think about this, if this hydrogen was drawn out here, then this carbon is on the same side as carbon number 1 allylic, and the hydrogen is on the same side as number 4 allylic. So in some sense they're a little different, but in terms of energy value, they're both second-degree carbons and allylic, and we're really going to assume there's not too much difference between those two guys, even though they might be cis or trans to this group. In this molecule down here, number 6 and number 3 are allylic, and again, because of symmetry, I can't really tell the difference between 3 and 6. So I've labeled which ones are allylic in here, and we've talked about how many are unique, and you need to be able to do that. Alright, so now that you know what we mean when we talk about an allylic position, then we're going to talk about how we can take advantage of that. And this reaction is only going to work with bromine because chlorine radicals are too reactive. If you recall, the strength of a chlorine-chlorine bond is more than the strength of a bromine-to-bromine bond, and that means that the radical that you create if you break the bond homolytically is not as stable. And so these chlorine radicals are so reactive that they're not very selective. And so even though the reaction works in principle, it also produces a bunch of other things, and so we won't be able to use it effectively if we try it with chlorine. It's not that there's no reaction, it's just not a useful way of going. And we don't also have a similar kind of compound like the one we're going to be able to use for bromine. We don't have something like that for chlorine. And in principle, we could do this reaction with bromine molecules since they can produce radicals, but we could get some side reactions like halogenation, and we don't want that. And again, we're trying to minimize side reactions and just get what we want. So we're going to use this molecule, which is called N-bromosuccinimide, or NBS for short. Now, we did see this in the previous chapter as a way to do halohydrin formation, and minimize side reactions with dihalogenation. And I told you we would be seeing that molecule again. Something else you'll notice about the conditions is that we're going to use light, and that's the symbol for light. And whenever you see that, that means we're going to do a radical reaction. Now we're not going to actually go through all the radical steps the way we just did in the previous section about free radical halogenation of alkanes, but you know that it's a radical process, so there's going to be steps involved. And this particular example, we again have a symmetrical molecule. These two positions are allylic, and I can't really tell the difference between them. And so if we replace one of these, any one of these allylic hydrogens with a bromine in this reaction, then we'll get this molecule. And so either of these hydrogens, we still get the same molecule. And by the way, anytime you're not sure if the molecule is really the same or different, you can name it. So if you had replaced one of these hydrogens down here with the bromine, you would number it 1, 2, 3, so it would be 3-bromocyclohexene, and that's the same thing as this. It's still 1, 2, 3 bromocyclohexene. So that's one reason that knowing how to name things is important, because then it can help you answer that question. Is this really the same thing, or is it different? Now your other side product out of here, because of the radical process, is succinimide, and we'll just have to get rid of that as a side product. And incidentally, if you were using NBS in a synthesis problem, remember I told you that you can use molecules whose carbons are not going to become incorporated into your product. Well, these carbons in the NBS end up over here, and that's not in the product. So you could use this molecule to manipulate this molecule if that was part of your problem, with no consequences. And so the next question we want to answer is, what is special about this allylic position? And we have to go again to the table of bond association energies to help answer this question. Now I've taken parts of the bond association energy table and put them together here. So this is not all the same column in the table in the book. It goes down to something like about here, and then this portion of the table is in another column. And we want to again look at CH bonds, because we want to see how the allylic position is similar and different from others. And so I've highlighted the ones we've been talking about in the past, which were the alkane type CH bonds going from quote-unquote 0 degree, primary, secondary, and tertiary. And then I have this one I've highlighted, and this one is a vinyl CH. And you notice that it's more than any of the alkyl type CHs. And then this one down here is allylic. So it's one carbon away from this double bond, and it's 369. And if you look at that, that's the smallest of all of these that are highlighted. So which one is easiest to break? Well, it's the smallest one, which is the allylic position CH bond, which tells you a little bit about why we're doing this. And you could also wonder about, well, what about the carbon-carbon double bond? Why isn't it getting involved? And we talked about that a little bit in the last section, that sometimes these reactions work on hydrogens because it's easier to get at the hydrogens because they're kind of on the edges of the molecule, as opposed to the carbon-carbon bonds, which are down inside. Admittedly, the pi bond is a little more accessible than the sigma bonds, but nevertheless, sometimes it's possible to do things with the hydrogens and leave the alkene alone, depending on how you run the reaction, and that's exactly why we want to use NBS this time. And so the next obvious question is, why is the allylic bond easier, CH bond easier to break? And it has to do with the radical. Remember, when you break the bond, the piece that you create, in this case we're talking about homolytic bond cleavage, so we're creating an allylic radical. And this is a very simple case, a simple example where we just have, this would be the molecule that was on the table. And so if you break that CH bond, you make this radical, and it can be resonance stabilized. So it could either be on this carbon or this carbon, going back and forth. Remember, this symbol means resonance. Or what you're really saying is that there's 2 electrons here and 1 electron here being shared by all 3 of these carbons. Now, there's a total of 3 electrons, and this is the book's picture, and they put 1 here and 1 here and 1 here in these dashed lines. But really, it's just a smear of 3 electrons over all 3 carbons, and in a sense, there's not really a whole lot of this picture or this picture. It's just really this smear of these electrons being shared by all 3 atoms. Here's another drawing. And you'll notice that all of those atoms, the hydrogens and the carbons, are all in the same plane in order for all those orbitals to be able to overlap. So now you can go back to your order of stability of CH bonds and carbons and radicals and kind of expand the series. So if you had, we had talked earlier about a methyl was less stable than primary, which was less stable than secondary and tertiary. Well, now you can say allylic is more stable than even tertiary, and vinyl is less stable than methyl. And again, we get all those numbers, and how we know this order is from those bond association energies. So how does that affect the products of the reactions? Well, we had this simple example from the earlier slide where we said if we used cyclohexene with NBS, we could make the bromine, and so we would have a 3-bromocyclohexene. And we have this example also where we just showed the resonance structure for this guy when it made a radical here, and so we can put the bromine wherever the radical is. And in these 2 cases, it's pretty straightforward. There's no worries. There's only one product that you can make. But remember that this radical is spending part of its time at either end, and this kind of color coordinated thing says that there's electron richness. In other words, the center carbon is always got its outer shell of 8, but there's some electron density missing from either end. So theoretically, the carbon on either end could have reacted with the bromine. Well, again, I can't really tell the difference between this position and this picture, which is the radical you get from here, but I may be able to tell in some cases because the allylic positions, if they do resonance, are not always going to lead to the same thing in both ends. We need to look at some examples of that. So let's go back to this molecule. Now we figured out which positions were allylic, and we also figured out that they were all identical. All the ones that we picked out that were allylic, we couldn't tell the difference between them. So if we start reacting this molecule with the NBS in the presence of light, and we go from any one of those to make a radical, we need to draw a picture of that. So here's a picture of, I've just picked number 6 and I made a radical there. Well, that through resonance goes down to here. So this is the other end of the allylic radical resonance structure. Now all you have to do is you take this 3-atom sequence and you flip. So involve the double bond and the carbon with the radical on it and just flip. And so that's what the other resonance structure is. So now you can put the bromine here or you can put the bromine here. And so we need to draw those products. And now you see they're not the same. We really do have 2 different products. Now it doesn't matter which one of these original allylic positions we picked off. We only get one actual radical stabilization, these 2 pictures, but 2 products are going to result. But that's all, just 2 products, because all of those allylic positions were identical. Alright, let's try a little bit more complicated example. So we had also talked about this molecule. And remember, I said if we don't worry about the cis or trans thing, we have 2 different positions. The one on number 7 is different than the one on the ring. Alright, so let's start looking at where we might have a picture. So let's say we just start up here and we take a hydrogen off of there and we'll draw that Okay, so we would have a radical here. Remember, you just flip down to the other end of the 3-atom sequence. So this is the radical from that. So what products are you going to get? We can either put the bromine on carbon number 5 or on number 7. Are those the same thing? No, they're not the same molecule. And we could go back to our original molecule and say, well, okay, what if we take a hydrogen off of one or the other of these and draw pictures of those? And I picked number 1, and remember the resonance is going to go like this, so there's the other end of it. And now we'll draw the products that we would get if we put a bromine on either end of that radical. And again, are these the same thing as each other? No. Are they the same thing as anything over here? No. So you would actually get 4 different molecules from this structure, even though there are only 2 different allylic positions. They are different, and you can lead to 4 different products, which is kind of a mess. So here's another example from the book. This molecule looks on first inspection like a fairly simple thing, just 1 octene. You put in these conditions and you can make the allylic radical. There's only 1 allylic position, but when it goes through resonance, the other end of the 3-atom sequence puts the radical down here on the end. So you can make products where you have some of the bromine here and some of it on the end. Now they actually gave you the percentage of the product that looks like this, and there's also some cis-trans stuff, so really you have 3 products. And again, it's kind of messy, and an obvious question is, why is this product dominating? And that is going to be because of it's the more substituted double bond, which is something we've talked about in another context. But the ultimately, you know, you're still going to get mixtures. And so we want to know, you know, what can we do with all of this information? One of the things is that you need to be able to find all the allylic positions and figure out how many of them are unique and how many are different. You also need to be able to draw the resonance structure. So once you figure out which are the allylic positions, pick off the hydrogen, you have a radical, draw the resonance structure of the other end of that radical. And then once you can do that, you can draw the products and you can figure out how many molecules you really are making. And so then the next obvious thing that you want to know is, well, when can we actually take advantage of this reaction without making a mess? And really it's going to be any cyclic alkene that's just unsubstituted, undecorated cycloalkene. So the example we saw earlier was cyclohexene. That only gave 1 product. Cyclopentene, cycloheptene, all of those just plain undecorated cyclohexene cycloalkene molecules will work. The only other thing that only gives 1 product was just propene, and that was also an example that only gives 1, and that's it. Another thing you might wonder is, well, okay, why did we want to do this? And I mentioned this to start with. We're keeping the alkene. So you're taking advantage of the presence of the alkene without losing it, so you're creating 2 functional groups. In Chapter 8, we talked about alkenes that we would utilize the alkene, and then we could make another functional group out of that, but we used up the alkene. The pi system electrons went into making some bonds to other things. And this lets us keep that pi system, that double bond, and you're still adding another functional group. So that's a little different philosophically. And so once you've got that halogen in there, that other halogen in there, then you could use that. And here's one way you could use it. So here's an example of a plain undecorated cycloalkene that works. We saw that one already. You can use the NBS and the light to make the 3-bromocyclohexene. And then you could dehydrohalogenate that. So you could put another double bond in there if you want, and we'll actually use that reaction, this series, in Chapter 14 to make dienes. But anything that you can do with the halogen, you can do now, and you still got the double bond. Some other things that we've done in the past to make alkyl halides, just by way of review, I mentioned this a little bit and I thought I'd just make sure you remember. We can make alkyl halides from alkenes, and that uses up the double bond. So we did Markovnikov addition and all the rules about that. We also did halogenation, so again we used up the double bond, but we did make two functional groups. We did hydrohalogenation of alkynes, and so we may use up one or both of the pi systems there, and get in more than one halogen, and this isn't all of them that incorporated halogens into molecules, but these are the main ones that make the point of you've lost the pi system when you did any of those things eventually. So so that is the end of this portion of this chapter, and in the book we're at the end of 10.4.